The overall goal of this protocol is to describe the use of photoactivatable peptide-MHC to induce polarized signaling in T lymphocytes.
This method can help answer key questions in immune cell signal transduction, such as how lymphocytes transduce rapid, localized signals and then mount polarized cellular responses to those signals. The main advantage of this technique is that it offers spatial-temporal control of T-cell signaling. It does this by fixing the precise time and location of T-cell receptor activation. Demonstrating the procedure will be Elisa Sanchez, a grad student from my laboratory.
Begin by adding biotinylated poly-L-lysine, or bio-PLL, diluted 1:500 in distilled deionized water to 8 well-chambered cover glass. Incubate for 30 minutes at room temperature. Following the incubation, wash with distilled deionized water and then dry for 2 hours at room temperature. Block bio-PLL-coated surfaces with blocking buffer consisting of heaps buffered saline with 2% BSA for 30 minutes at room temperature. Following the incubation, remove the blocking buffer from the bio-PLL-coated surfaces without allowing the wells to dry. Then add streptavidin and incubate at 4 degrees Celsius. After an hour, wash the coated surfaces in HBS. Fill and invert the chamber slide wells 4 to 5 times, removing HBS from the wells. Do not allow the wells to dry. Add the specific biotinylated photoactivatable peptide major histocompatibility complex ligands and adhere.